Mirava system, and today we'll move to the MINFLUX system. So actually, after this 1 hour presentation, and we are going to have 30 minutes pause time, and then my colleagues Evelyn and Nichols is going to demo the MINFLUX remote demo from Germany. It's a live demo. So I will start the presentation now. Okay, what you see on screen now is the big giant MINFLUX. It's a huge one. It's the first commercial system with molecular size resolution. And now you see the resolution formula, as you can see here, it's something like air divided by some Fermi numbers. So the concept is totally different from STAD, from confocal microscopy, and from all the others. So as an introduction, I will start with the overview of the super resolution, same as 2 days ago. So I studied neuroscience. I use a synapse as an example. So these are the structure in the synapse and the vesicle size, for example, 40 nanometers. And these structures cannot be resolved by confocal microscopy because the XY resolution is around 200 nanometer. Everybody knows the diffraction barrier from the light microscopy. And with our STAD microscopy, the XY resolution comes down to 20 nanometer, and even with our MINFLUX microscopy, the resolution is around 2 nanometer, and we can do a lot of interesting research. The resolution improvement has dramatic impact on the scientific data authenticity or the research quality, as you can see here. From the confocal, it's misleading data, right? You see colocalization, you might think there's a translocation of the proteins, while instead it is so clear the pre and the post synaptic compartment, they're apart from each other. So actually there's no interaction of these two proteins being imaged. And that's the power of the super resolution optical microscopy. Talking about super resolution, we have to mention Stefan W. He's the first one in the world who has the concept of super resolution optical microscopy. And he shared the Nobel Prize in 2014 in chemistry. He invented STAD, he invented MINFLUX, he's the founder of Abberior. And actually, as I explained to you 2 days ago, we have two companies. One is called Abberior Instruments. We produced a lot of nice nanoscopies. Another one is called Abberior Labeling and Dye Technology, because you think the resolution improves, or the labeling technology and the complex size and positions, all these you have to take into consideration in your experimental design. And also, as I mentioned 2 days ago, that from our STAD microscopy, we are the only one in the world who produced absolute raw data by photon counts per pixel directly from the detector. So from our STAD machine, there's no post-processing needed to generate a beautiful image. We produced absolute physical data. We're so confident with our data quality. And as you are going to see from today's live demo, we use nuclear pore complex as benchmark to test our microscopy, because it is a very good resolved structure by the electron microscopy. And it is so important. There are so many proteins are involved in this structure. There's sensor proteins, scaffold proteins. It's sitting across the nuclear membrane, outside and inside. And as shown by the electron microscopy, you see actually there are 8 subunits of the nuclear pore complex, symmetric in the XY view. And in the Z, it's asymmetric, right, as you can see here. All these research were done by the electron microscopy. Why? Because the diameter of the nuclear pore complex is less than 200 nanometer. In other words, the conventional optical microscopy cannot resolve the structure. If you would like to image a nuclear pore complex by, for example, a confocal microscopy, you cannot see it clearly, as you see here on this slide. This is Nobel Prize 2014 chemistry official slide. They use Abberior Instruments STAD to show you the 2-color 2D nuclear pore complex imaging. As you can see, the STAD microscopy, the resolution is 10 times better than the confocal. It can show the 8 subunits clearly in the XY view, while the green protein is the central protein of the nuclear pore complex. And actually, here is just 2D. Actually, we know from the 3D, the central protein is underneath, right? It's in the inner part of the nuclear membrane, while the 8 subunits, they're sitting across the membrane. So also you have to keep in mind 3D is also important. In that year's Nobel Prize were awarded to 2 technology. One is STED microscopy. It is based on a scanning microscopy, for example, confocal. Another technology is single molecule localization based method, for example, palm, storm, or some other words, right? It is based on a camera, it is based on wide field camera system like TIRF, total internal reflection microscopy. So both techniques, STED and single molecule localization, they can reach the resolution around 20 nanometer, like 10 years ago, in the XY. That's in 2014. Then in 2017, we launched the MINFLUX technology. We still use nuclear pore complex 96 protein as an example. Learned from the electron microscopy, nuclear pore 96 protein, there are 32 copies of these molecules in the whole nuclear pore complex. In each of these 8 subunits, there are 4 copies of this protein. 2 are outside memory, 2 are inside the memory. This is resolved by the electron microscopy. We can also measure the diameter and distance between the subunits. However, with the MINFLUX, which is an optical microscopy, we can do the same thing, as you can see on this screen. We have very similar localization distribution of that molecules, right? If you measure the distance, we can get a similar data as the electron microscopy. In addition, while the sample preparation in electron microscopy is a kind of lousy, it's a dead sample, it's a very complicated, right? You lose a lot of biocharacters during the sample preparation. While in the MINFLUX, you can preserve all those biocharacters. Because MINFLUX is an optical microscopy, you can do the sample preparation in fixed cell, in live cells, in 2-color, in 3D later. We can also do the single molecule tracking in live cells, while the resolution is coming down to 1 or 2 nanometer. So it is a very good complementary to the electron microscopy. While the electron microscopy shows the structure of the proteins, the MINFLUX can do a lot of functioning research on the single molecule level. And now you can compare the resolution from confocal of the nuclear pore complex, 200 nanometer. You can see no details. STED is 10 times better than the confocal, while the MINFLUX is 100 times better than the confocal resolution. And this is a review paper published in 2017. It shows all the PSF, point spread function, of all kinds of optical microscopy. What does point spread function mean? It shows you the smallest light dot size and shape in each kind of optical microscopy. The up panel scale bar is 200 nanometer. We use confocal as a reference. XY is 200 nanometer. Z is even worse. It's 500 nanometer. And everybody knows this is true in the optical microscopy. It's like elongated American football size and shape. And however, confocal method is pure optical. All the other method in the up panel with the stripes, you need mathematical calculation, post-processing, to calculate the resolution, to improve the resolution 2-fold is better than the confocal, like deconvolution, error scan, 2D sim, 3D sim, lattice sheets, and so on. So the lower panel, the scale bar is 50 nanometer. As I said, STED is pure optical method. In our STED, we do not need any post-processing to generate a STED image. So this has been demoed 2 days ago. From our STED system, the XY resolution, the best XY resolution is around 20 nanometer. And for the 3D STED, we can do isotropic resolution in X, Y, and Z around 60 nanometer. This is one of the Nobel Prize technology. The another one is single molecule localization, like Palm and Storm. Via the localization calculation, you got the XY localization precision, or let's say resolution in a common word, around 20 nanometer. And the 3D resolution is in a similar range as our isotropic optical stat, a little bit worse. And the MINFLUX is actually the combination of the two Nobel Prize technology. I'm going to explain later. So on this slide, listed all the super resolution method. We always use confocal as a reference. Confocal is a very general, common instrument now. I use first time for me to use confocal was in 1997. Like at that moment, confocal was absolutely high-end. Now, confocal is a very, let's say, entry-level technique to do the biological research. Now we have 3 super resolution or high resolution catalogs for super resolution. The first one is structure related, either at the illumination side or at the detection side. Structure illumination, SIM, or at the detection side like spinning disk, array detector. These structure-related methods can boost the resolution 2-fold better in X, Y, and Z compared to the confocal. The second category is single molecule localization method. Palm and Storm 10-fold better than the confocal in X, Y, Z, and our Abberior MINFLUX, which is 100-fold better and very fast, 100-fold faster than camera-based single molecule localization. The third category is STAD. STAD is also 10-fold better, the resolution compared to the confocal in XYZ, and is the only super resolution method with direct physical output, easy to use as a confocal. No mathematical calculation or post-processing is required for STAD super resolution. And from our company, we have 3 kinds of nanoscopy. The up to their STAD technology. The last one is this big giant MINFLUX. The principle, they are different. So from the STAD, we have 2 models. One is an entry level, it's a 2D STAD. We call it Steadicon. As I explained to you 2 days ago, any STAD microscopy, it is intrinsic optical perfect confocal. Otherwise, it cannot reach the super resolution. So for the 2D STAD, we have the name Steadicon, STAD and confocal. It's the second generation. We integrated the film, lifetime imaging into the system. Anytime you test it with beads, optical raw data without any post-processing, we can reach 2D STAD resolution 30 to 40 nanometer. Anytime, anywhere with the Steadicon. Then in the middle, we have the giant STAD system. It's called Mirava. It's an all-in-one system. You can do millimeter scale imaging down to single molecule level imaging. It's a combination of confocal technology, 3D STAD, matrix array detector, 2D MINFLUX, and a lot of other modules as we demoed 2 days ago. So the 2D optical STAD resolution is 20 to 30 nanometer on the Mirava, and the 3D isotropic optical STAD resolution 60 to 80 nanometer. While on the Mirava, we have a simple 2D single color MINFLUX module. The localization precision is 3 to 5 nanometer. And today we're going to talk about the giant big MINFLUX and do the demo. On that big MINFLUX, the 2D localization precision is 1 to 3 nanometer. 3D localization precision is 3 to 5 nanometer. And we can do single molecule tracking in live cell, 100 microsecond per data point. This is very, very fast. If you know how to do the storm, for example, you use the camera exposure time like 10 milliseconds, right? 100 frames per second, it's 10 milliseconds. Why? Because you have to capture the blinking of the dye. The duration of the blinking of a dye is commonly ranged from 10 to 30, 40, 50 milliseconds. That's why you have to expose your camera at 100 frames per second, so you will capture the blinking process of that dye 2 or 3 or 4 frames on your camera for the further calculation. Now with our MINFLUX, we can point one single molecule in 100 microseconds, which is very, very fast, 100 times faster than any camera-based method. Okay, to talk about the MINFLUX principle, we have to have some knowledge, right? So it's a combination of the STED and single molecule localization. So I would like to show this slide, which I showed 2 days ago, to prepare you for understanding the MINFLUX principle. And this slide is called STED intensity and resolution. In a normal confocal microscopy, we have a Gaussian illumination pattern. As you can see here, it's a Gaussian pattern. Due to the diffraction barrier, the diameter of that pattern, the smallest one is 200 nanometer. Practically, it's 230, 250, right? You can do nothing with it. Then we have the STED donut beam deployed and overlapped to the Gaussian excitation beam. And if you draw a cross-section, the line profile, you see the energy distribution, excitation energy distribution of the STED donut. It's shown in this red curve. It overlaps to the green curve at the back, which is a normal confocal excitation Gaussian beam. In the overlap area, there's STED effect. Therefore, on the detector, it's dark. Only in the donut center, there's no STED beam. Therefore, you have the normal confocal excitation. The combined effective spot size is getting smaller. Therefore, the resolution improves. And if you increase the STED power more and more, the central donut is confined less and less. Therefore, the resolution is improved more and more, right? That's the STED principle. That's why we said we have to use very good dye. If that dye can handle a lot of STED power, then you can get better resolution. Okay? Now, this is STED principle. Actually, in MINFLUX, we only have one beam. We only have the excitation beam. And we prepare the excitation beam in a donut shape. Okay? We prepare the excitation beam in a donut shape, and then we increase the intensity of the excitation beam. Since it's a donut shape, as you can see on this slide, the center part is getting smaller and smaller. So the diameter, you can have that concept in mind. The diameter is getting shorter and shorter. Okay, this is the first thing you should learn in your mind. Then the second, you have to understand what single molecule localization is. As this slide shows you, actually, we generate a picture by a lot of dots, by a lot of coordinates of the dot. It's not like confocal. It's not like STED. Confocal and STED is painting with brush, right? While in the single molecule localization is painting with points. It's pointillism. So actually, from this kind of microscopy, Palm and Storm and our MINFLUX is the same. The raw data is a lot of coordinates of the dot, and then you reconstruct them into one picture. However, the Storm or Palm, they use camera-based method, and it takes half an hour to collect thousands of frames with all the random dot, right? Then it takes another maybe half an hour to calculate the position of each dot, or to do the mathematical correction of the shift or the distortion of the light, then to calculate the position. So it takes a long time for you to generate one image in palm and storm, these kind of technology. And only after that long time, like 1 hour, it's your first time to see that image. So this is a restriction in the application of these technology. It takes too much time to get to a very nice image. And the MINFLUX is a combination of these two. And we, again, start with the example of the neuroscience. I studied neuroscience like 26 years ago. I studied Alzheimer's disease. Like 26 years ago, I studied Alzheimer's disease. We mainly focus on the microtubule in the axon. I studied axon transport and how the neuronal process, they generated the polarity and so on, and degenerated in the Alzheimer's. In those years, we know nothing about the organization of actin in the axon. We know there's a lot of actin in the somatodendritic compartment, in the axon initial segment, but we don't know how the actin cytoskeleton was organized in the axon. So this was the first paper by using very complicated dual objective storm method. It's a methodology paper published by Xiaoyu Li, who invented actually storm in Science in 2013, and tried to resolve the structure of the actin and its capping protein called adenosine and the scaffold protein called spectrin, how they formed periodic arrangement in the actin in the axon, neuronal axon. So it's a methodology paper published in Science in 2013. Now, while the technology, they are much more improved, as I already showed, with our Mirava, by simply scanning down as a confocal scan in the STED mode, you can already imagine easily the two-color 2D periodic arrangement of the spectrin protein and adenosine. And with the MINFLUX, we can even do the single molecule imaging. While in confocal, actually, it's a very basic technology. You cannot resolve any details with the confocal microscopy. Please keep in mind the sample preparation knowledge, effort, cost, imaging efforts. They're identical in confocal and in STED. And why not do everything in the STED? Because it will give you much more information compared to confocal. Now we come back to MINFLUX. As demoed already 2 days ago, on the Mirava STED platform, we already have the MINFLUX module, and it is so easy to use. These are the ER single molecule imaging. On the left side is the confocal raw data. As I told you, we only show the raw data and by the photo counting. Okay, you see the maximum, we have 149 photons counted in that pixel. Okay, there's no intensity concept in our system. It's absolute true photon numbers. And on the right side is the single molecule imaging of that proteins. And actually, it's so easy to handle. The students can handle it so easily. So this is single molecule imaging of the MINFLUX on the Mirava platform. And this example, I also explained, we had the China Cell Biology Conference 3 weeks ago. We delivered a demo Mirava system in the conference room, right? What you see here is the system being put in the conference room, and we are trying to do single molecule imaging. And in the middle, you see the stabilization system of our MINFLUX. You are going to see it in the following demo. So it works in the astron level, 0.3 nanometer in XY and 0.9 nanometer in the Z direction. So it works in the astron level to stabilize the whole system. And under that condition, we do the single molecule imaging That customer brought his sample. It's a PSD195 protein, post-synaptic density 95 protein, right? We measure the diameter of that one. It actually ranges from 6 to 11 or 12 nanometer. The example showed you here is 7.18 nanometer. That's the mini MINFLUX module on the Mirror. Then we have the giant MINFLUX. This data came from our first MINFLUX customer, EMBL, EMBL in Heidelberg. The first system was installed in 2020, August. Two colleagues from Abberior and we together installed the system in EMBL. So now EMBL actually purchased the 3 big MINFLUX. So the first thing they did is to imaging the spectrum stripes. And they try to see if they can see the same thing as Xiaoyu published. Because Xiaoyu published by the very complicated storm method, right? In the science, it's the first time human beings saw that structures. Now with the MINFLUX, they can easily see the organization of the spectrum proteins in the axon. The localization precision is less than 2 nanometer. However, you see on this slide, you would doubt whether this is periodically or arranged or it's a kind of helix structure, right? Either it's truly helix or the axon was somehow tilted. Then you see from an angle, that gives you a kind of feeling it's a kind of helix. This can be further evaluated by the 3D MINFLUX. This is the neuron and confocal. We imaging that axon, and from the 3D MINFLUX spectrum protein imaging, now we see clearly it's periodic, organized. Pseudo color indicated different Z layer. It's rotating now in the XZ view, right? So now the 3D MINFLUX can show you a kind of isotropic resolution in X, Y, and Z less than 3 nanometer. Okay, the first MINFLUX paper was published in Science by Stefan W in 2017. So actually MINFLUX, the concept is single molecule localization. It's the same as STORM and PALM. However, it utilizes a donut-shaped excitation beam, as I told before. It's not steady. Instead, we have 2 beams. Steady beam is donut-shaped. Excitation beam is Gaussian. That's steady. In MINFLUX, you only have 1 beam. The excitation beam is made in a donut shape. And through a very sophisticated method, movement, and calculation, then we have 100 times better the efficiency compared to the camera-based PALM and STORM, those single molecule localization technique. And then we can reach the truly single digital, single molecule level imaging. In that paper, we use DNA origami as an example to test first. We can control the DNA origami, the dimension, the size, right? For example, in this example, we use 9 molecules and we arrange them in an 11 or 12 nanometer array. And 1 molecule was switched off. Therefore, you see no signal in the MINFLUX microscopy. While we try to localize the coordinates of every molecule, every other molecule, 8 molecules, right? So as I said, single molecule localization, it's a kind of, the raw data is a coordinates, okay? We try to localize several times for 1 molecule. It's similar like in Olympic shooting game, right? You try to shoot a target multiple times. If all the bullet holes, they spread widely, it's a not good system, right? If all the bullet holes, they are more concentrated to the center, then your system is very accurate. We do the same, right? All the bullet holes from the MINFLUX, actually they are all the coordinates, try to tell where the molecule is. If you do a fitting, you see actually the deviation is around 1 nanometer, right? The first group of the bullet holes, deviation is 1 nanometer. Second group, 0.8 nanometer. Third one is 1.3 nanometer. That's the localization precision, okay? That's actually the resolution normally we say in localization microscopy. Actually, there's a factor 2.3 to the normal full width half maximal concept as we learned from other microscopy. Okay. To give you an idea how accurate it is, do you know what kind of size a GFP molecule is? Green fluorescence protein. A GFP molecule is around 5 or 6 nanometer, this size, okay? Now you see our accuracy is around 1 nanometer. It means from the MINFLUX, it tells you, okay, your GFP molecule is at this coordinates in XY. The deviation is within 1 nanometer. Okay, the molecule size itself is more than 5 nanometer, so actually it's very accurate. With that accuracy, now you use the center of all the bullet holes as a representative point to the next center, to the third center, you measure distance 11.7, 11.6. It's almost identical to the reality. That's the MINFLUX. To further explain the principle of the MINFLUX, we use the GPS satellite as an example. If you only have one GPS satellite to tell you, try to localize where your car is, you have very low precision. If at the same time there are several satellites depending on the relative distance or position, and then they try to calculate where your car is, and then you have very high precision. Actually, we do the same thing in the MINFLUX. So MINFLUX is a single molecule localization technique. So you have to somehow to make the dye blinking, right? Either by the conventional storm buffer or by the DNA painting, right? DNA painting is actually very matured now. We're going to demo you with DNA paint. We have a lot of product from the DNA painting from us. So during the single molecule on state, so in that state, the molecule is blinking, right? What does that mean? It means in a certain time, in a certain area, there's only one molecule is on, is blinking. You can see the signal, okay? At that moment, we try to hit that molecule by the center of the excitation donut. Okay, as you can see here, boom, the microscope try to deploy the excitation donut, try to hit it by the center. If we are so lucky, then the center of the donut of the excitation will hit the molecule, right? Then you know already from the stat, in the donut center, there's no energy, there's no excitation beam. So actually you see no signal on the detector. Actually, we are not that lucky all the time, right? So there will be a slightly deviation from the center of the donut to the actual position of that single molecule. There's a little bit deviation. Then, as you can see here, we have a very low excitation power, resulted in very low signal photon. For example, 3 photons. Okay, this is true data in that paper. So actually, our detector is true single photon counting detector. I explained you already in the stat part, right? It's not claimed the single photon, it's true single photon. Okay, that's the first satellite position. Okay, then we move the donut, excitation donut, to a next position. So the center is a little bit further away from the reality of the reality position of that single molecule. Then you have a higher excitation power. Then you have higher signal photon counts. Then based on the mathematics behind, we move to a third position and a fourth position. And based on the signal photons and related to their position, we can calculate the exact position of that molecule. Okay, we calculate the position of that molecule. This is one thing. There's another thing we have to be sure your calculation is accurate. So these are two different things. One is to calculate the position, and you are not sure how accurate it is. Then actually the most important thing is the accuracy of the system. It is controlled by this formula. Okay, it's air, which is a kind of distance, and n is all the signal photons we count. And in x, it's air divided by square 16n, and xy, it's even it's worse than one axis, right? It's easy to understand. So the formula behind the accuracy formula, it's actually Kramer-Rao bound. Kramer is a Swedish mathematician. Rao is an Indian US mathematician. Kramer-Rao bound described the best estimation, non-biased best estimation in statistics. In other words, if the accuracy or precision or resolution defined by this formula, then it's the best one under that kind of circumstance. You cannot be even better, okay? This is the MINFLUX concept. Why it's very important? Because in all the other microscope, you try to get as much as signal photon, right? You want to have the high signal noise ratio. You use the Gaussian maximal, try to excite your photons. And then you have all the problem like bleeding, like vibration, like all these kind of things. You have to fine tune your storm buffer. While now in the MINFLUX, it's so clever. Once the donut center is getting close to the molecule, the signal is darker and darker. While it's away, then the signal is brighter. It's a very clever way, right? So you know, okay, the signal is getting darker and darker. I'm in the right direction. In this way, you protect a lot of signal photons being wasted, or being bleached, okay? And in the first paper, you see the donut was moved 4 times, right? 3 positions and 1 original position. One cycle of this movement is around 100 microseconds. So actually, it's very fast. One cycle, 100 microseconds. You can already get a localization precision. And then if you see the formula, you see there are actually 2 parts. One is the signal photon, one is the air. In storm and palm, you only have the signal photon number, right? Therefore, based on the mathematical calculation, if you get a lot of signal photon, the accuracy is better. However, in our system, we have air. By controlling the air, the efficiency of the whole system is much increased. We can do a very simple calculation. If we control the air, the initial air at 100 nanometer, if we want to reach 1 nanometer accuracy, roughly you need 10K signal photons, right? 10K signal photons, square root is 100. 100 divided by 100, it's 1 nanometer. That's the initial air. Okay, if we reduce the air from 100 nanometer to 10 nanometer, for example, and then we still want to reach the 1 nanometer accuracy, and that circumstance, you only need 100 photons, signal photons, right? 100 signal photons, square root is 10. 10 divided by 10, it's again 1 nanometer. So by controlling the air from a large air, then you reduce the air to a small number, then the requirement to get the signal photon total counts are much reduced. That's why the accuracy is very high. And actually, in our system, we do several iterations. We can control the air, for example, 100 nanometer at the beginning. We want a localization precision, for example, 10 nanometer at the beginning. Then you calculate how many signal photons you must get from the system. And actually, the system is running automatically. It's a turnkey solution. Then once the first signal photons, the number they reached, okay, you know, I'm now at 10 nanometer localization precision. Then we do the iteration. We reduced air from 100, for example, to 80. Then we set the accuracy from 10 nanometer, let's say, to 7 nanometer. Then you calculate the requested signal photon numbers. If you meet that condition, you can do the further iteration to reduce the error, to set the accuracy even higher. Then normally after 4 or 5 times iteration in our system, you will always reach around 2 nanometer localization precision. That's the whole behind the MINFLUX. And actually, it's automated. It's turnkey solution. You do nothing about all the steps I described. Okay. So from the mathematics, as you can see, there are 2 parameters. One is air, one is n, right? We always try to control the air. We do not try to increase a lot of n. Why? It is because of Poisson noise. What does Poisson noise mean? It's the major noise in all kinds of microscope. The Poisson noise, the range of the Poisson noise is related to the signal intensity. In other words, if you have a high fluorescence signal intensity, the range of the Poisson noise is large. If you have a low intensity, the range of the Poisson noise is small. If you have a large Poisson noise, the mathematic fitting is not accurate. It will be very difficult to detect. As we go to the temple, we light a candle. There are so many candles. The signal intensity is very high. Then you light one candle or you put one candle distinguished. It's difficult to detect because the noise range is very high. If we go to a very nice restaurant, you light 2 candles or 3 candles. The total signal intensity is low. Therefore, it's very sensitive if you have one candle lighted up or distinguished. The Poisson noise range is smaller. It's more sensitive for the system to detect. Okay? Then if we do the mathematical fitting, as you can see, the Poisson noise actually will decide how the accuracy is. Therefore, if we try to reduce the air, right, as you can see here, try to reduce the air with the same Poisson noise level, the distance sensitivity in the x-axis is more and more sensitive. Okay, right? It's easily explained in this slide, right? If we try to reduce the air with the same signal background, the sensitivity of the position change in the x-axis is more sensitive. How to reduce the air? I explained already in the STED. You just increase the laser power, right? Instead, you increase the STED laser power. And in MINFLUX, you increase the excitation donut laser power. The air is getting smaller and smaller. That's why we do the iteration. With one large air, then we move to small air, even further small, and then the accuracy is even improved and improved to the maximum one of that system. It's always around 1 to 2 nanometer if you prepare a sample very nicely. Okay, in the commercial system, we move the donut actually into 7 positions, right, as it shows you here, to make some redundant. And then in the 2D MINFLUX, we have a 2D donut shape. It comes from our STED technology, right? We use SLM, spatial light modulate, to generate perfect 2D donut. I explained in the 2 days ago in the STED presentation. Then we start with a large air, and then we calculated roughly the position of that single molecule. Then in the second iteration, we make the air smaller and improve the accuracy. right? Then we do several iterations. The area is getting smaller, smaller, the accuracy is increased. This is 2D. In 3D, it's the same. We use SLM to generate a perfect 3D donut. It's the same as in the stat. That's why you can think how good our stat it is, right? Everything needs to be perfect. Practically, it needs to be perfect as theoretical numbers, right? And then in the 3D donut moves to these 1, 2, 3, 4, 5, 6, 7 positions, right? 4 are on the equator of the sphere. 1 is center, 1 is in the North Pole, 1 is in the South Pole. So this one is jumping here, 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 there. Here again, we used deformal mirror, as I explained to you in the Mirava presentation, to make the perfect 3D movement. Okay. So from the instrumentation level, from the technical level, it is very high-end, cutting edge. Actually, it's the top. Nobody else can use these kind of things. Then again, the same. We start with a long radius, then make it smaller and smaller. And now Stefan here is very happy. He said, my stat is 10 times better the resolution than confocal, while my MINFLUX is 100 times better. So actually, this slide shows you the relationship of the magnification and the resolution. Actually, it is very important. A lot of people are trying to mix these two concepts. These are the two different concepts, but they are related to each other. If you have a very good magnification, but poor resolution, no matter how large you magnify, you cannot see the details. On the other way, if you have very good resolution, but you do not magnify them, like in the confocal, you can tell this confocal, it's beautiful, super resolution, but you do not magnify them, you can still not see the details. So if you try to tell this is a super resolution system, you have to magnify the region of interest, then see the details. These two factors you have to keep in mind. So actually, as you see here, MINFLUX will magnify to a single nuclear pore complex, then you see all the details of the single molecules. That's the true super resolution. Okay, this slide I showed you, this is the 2D MINFLUX. We can do the 3D MINFLUX. We use nuclear pore complex as an example. As you are going to see in the demo, we can check with eyepiece to find the region of interest. That's a confocal. Then you click run. It's a automated turnkey solution. Now you see 2 layers in the 3D of the nuclear pore complex, that NUP96 protein, and then rotated in the XZ, XY view to see that molecule's position in the 3D. Then here's the early paper try to prove the methodology of a 2-color 3D MINFLUX. They try to resolve the mitochondria MIC60 protein, which is orange, and the ATP synthesize, which is blue. So they are apart from each other, so you can see clearly these 2-color 3D single molecule imaging. And then later, actually the authors try to resolve the MIC60, MIC10, and MIC30. These complex, they are very close to each other. That's why he first tried with 2 molecules that are apart from each other, try to prove the method. Then later, the MINFLUX is so welcome globally. We have a lot of installation of the MINFLUX. In Asia Pacific, we have now close to 10 big MINFLUX, or in the US or Europe, even much more. Then there's a lot of very advanced cutting-edge research done by the MINFLUX. Because of the time, I'm just trying to explain you actually several papers. The first one is actually my favorite paper. It's Piezo 1. There are 5 Nobel Prize technology related in this paper. Adam, he won the Nobel Prize in physiology and medicine by studying the piezo ion channel, which is a sensor on the cell to send the mechanical force. And Stefan here, we provide the MINFLUX, and the sample preparation is click chemistry, which is a Nobel Prize in 2022. So you insert just 1 dye to a certain amino acid position. Then we have the cryo-EM. We have the alpha folder. Both are Nobel Prize technology, right? From the cryo-EM and the alpha folder, They're the same. They show you a dead sample, the distance of the ion channel. While in the MINFLUX, it's a dynamic range, right? As I said, that's the power of the MINFLUX. The resolution is coming down to the EM level, and somehow the sample preparation is in a biological character preserved way. So you can test the range of that ion channel. Well, it's closed and open and closed and open. And then in the middle, then you try to destroy the membrane by the detergent. Then you see the range is getting smaller. And then you can add antagonist to make it open again. Then the range, the distance is open again. Then actually on the right side, the customer try to resolve the conformation trends of that single ion channel in live cells. First, they put the dye at the tip of that ion channel. You see the shape of the ion channel. It looks like a blade, 3 blades, right? So he first put the dye to the tip of the blades. He thought, okay, why the ion channel opens and closes, then actually the blade is stretching out and back, out and back. So there should be distance, measured different distance of that dye, but actually nothing happened. Then he moved the dye to the arm position of the blade. Then there's change, right? Resolved by the MINFLUX, 3D MINFLUX, actually there's change. It means while the ion channel open and close, it's not the blade stretching out and coming back. It's the elbow, it's lifting and put down, lifting and put down. So it's the first time to resolve an ion channel in the physiological conditions in live cells. It's a Nature paper in 2023. Just 3 years later, he published again in the Nature Piezo 2 by using the MINFLUX, 3D MINFLUX. Actually the same concept, same experimental procedure. It's so easy. Piezo 2 is expressed in somatosensor neurons, right? Then he did the same experiment as Piezo 1. You try to measure the distance, this and that. Then actually the most important thing is he found Piezo 2 is actually anchored to the actin cytoskeleton via filament B. Okay, it is different from Piezo 1. Piezo 1 is a kind of free in the cell membrane. That's why in the tracking data on the right top, it's the tracking of the Piezo 1. It's a kind of free movement. It sends all the direction mechanical force from all the directions in the cell membrane. While in the Piezo 2, it anchors to the cytoskeleton. So if you track the Piezo 2, it's more concentrated, as in the right middle panel. It's more concentrated. Then they find the domain which is responsible for this anchoring and try to mutate it again. Then the Piezo 2 is similar like Piezo 1. It's free movement in the cell membrane. So what does that mean? The Piezo 2 actually in the physiological condition, it anchors to the cytoskeleton, actin cytoskeleton beneath the cell membrane. It functions as a trampoline. That's why it sends the mechanical force in the Z direction. While Piezo 1 is free movement in the cell membrane, it sends the mechanical force in the XY direction. Okay? It's very advanced research, right? So then there are several other papers. Because of the time, I'm going to skip. Then we try to see another important function of the MINFLUX, which is tracking. Okay, we say MINFLUX, there are two functionality. One is called imaging, one is called tracking. Our tracking speed is also very fast, 100k hertz, which is 10k hertz, which is 100 microsecond per data point. As you can see here, we track a dye in a lipid bilayer. Again, I'm going to explain you some research done by the MINFLUX tracking. The typical one is tracking of kinesin, right? I studied in neuroscience. I did exactly this experiment almost 30 years ago, right? 25 years ago, we try to see the axonal transport of the kinesin. Kinesin is working on the microtubule by hydrolate the ATPs and two motor proteins and working on the microtubule, taking the cargo there. However, in the past 35 years, we can only use in vitro method. Because in vivo, it's not possible. The kinesin is working so fast and the signal is so weak, you can never detect it. And in vitro, how did we do? We polymerized the microtubule. We put the recombinant kinesin onto it. We can lower the ATP concentration. We can lower the temperature to make it move slower. And we put a very bright quantum dot on the top of the kinesin. Therefore, the camera can see it. And then since the quantum dot is so heavy and large, we have to use optical tweezer to hold the quantum dot for the kinesin during the movement. Then we use TIRF to see the single molecule working on the microtubule. With this method, in vitro method, there's a lot of publication in the past 35 years. High-level publication. The latest one was in 2021, even in Science. Try to resolve the working of the kinesin. So somehow it's quite clear by the in vitro study. Now with our MINFLUX, we can do the kinesin tracking in live cells. Okay, we have, for example, two publications on the same issue of the Science in March 2023. One is by Stefan W, one is by EMBR. EMBR is our first MINFLUX customer. The system was installed in 2020 August. The first science paper was 2023 March, right? You think how fast it is because it's very advanced instrument. Then it can really do a lot of fancy research. Okay, let's see the experiment. First, we mimic the in vitro experiment. We lower the ATP concentration at 10 micromolar. Then, by the tracking, we can see it is 8 nanometer sub-step, right? It is inconsistent with all the previous data, in vitro data. Now we do the physiological concentration of the ATP, which is 6 millimolar, magnitude high. Now you see the kinesin is moving very fast on the microtubule. It's working on microtubule 2D, right? You check the time resolution. Less than 1 second, we trace all the steps, right? 780 milliseconds, we trace all the steps. And again, shows 8 nanometer sub-step. While you see the later part of this tracking, actually, it's a kind of math. Why? Because the kinesin switched to another microtubule. Through the 3D tracking, you can resolve the trace of the kinesin. Actually, it moves on one microtubule and try to switch to other microtubules, either by diffusion to another microtubule, then try to work again. So this is the power of our MINFLUX microscopy. Another paper is even more interesting, right? So in this paper, the authors try to label the kinesin stalk with one dye. Okay, in the previous paper, the dye was labeled on the kinesin motor. In this paper, it's on the stalk of the kinesin. Then it's interesting. If we try to resolve the step size, it's 6 and 10, 6 and 10. It's not 8 nanometer, it's 8 minus 2, 8 plus 2. What does that mean? It means while the kinesin motor is moving, is working on the microtubule, the stalk has a kind of conformation change. Forward, backward, either rotating forward or rotating backward or move forward, move backward. This we don't know, but we see there's conformation change. It's 2 nanometer. So it's 8 minus 2, 8 plus 2, right? Try to prove this. We put another dye at the opposite part of the stalk, right? Then it's balanced again. We take these two dyes as one entity, then 8 nanometer is restored. So this is the first paper try to resolve the kinesin conformation change in a living cell, a science paper. Then later, actually, there's a lot of research has been done with the MINFLUX on the dining retrograde transport and so on. So a lot of transport tracking experiment are applied to a lot of kind of research. For example, in this paper, and you are going to hear from Evelyn during the demo, they use two-color MINFLUX imaging and tracking, try to see the transport impulse alpha, how it enters the nuclear pore complex. So it's a kind of 3D, right? From 2D, you see it getting close to the nuclear pore complex, and from the 3D, you see how it enters the nuclear pore complex from the cytosol into the nuclear. So actually, this is the last video of my talk. It's our promotion video of the MINFLUX. So once we do the imaging, we do the iteration. We increase the laser intensity and the air is getting smaller. Well, we can also do tracking, right? This is tracking. So this is the single molecule tracking of a dye to print out company logo, right? So actually the system is very capable. That's all of my talk. Thank you very much for your time, for your attention.
Xiaoyu said, I am the Senior MINFLUX Application Specialist for Europe, and Nichols is our Product Manager for MINFLUX system. And today we'd like to show you a little bit of how the system runs, and we'll also talk to you a little bit more about the different ways that the system can be used. So I would hand over now to Nichols directly at the microscope. You can see it in the camera there, and we'll take a look at what he's been running. Nichols, I think you're muted now.
Do you hear me?
Yeah.
Wonderful. Oh, headphones always producing problems. I hope you see my screen now.
We do.
Yes. Perfect. What you see here now is our MINFLUX inspector software that controls our microscope and a running measurement. I will now talk a bit about the different parts we have and how we do this. And then we set up another measurement. And I will show you how easily this is done in the beginning. So first thing you see a lot of open windows. Our system is a confocal microscope. So you can do normal confocal scanning. You can image cells or tissues or whatever you want in a normal confocal mode. And then you can just decide for a region that you want to take a look at and switch to the MINFLUX mode. This is something I've done before. We are looking here now at nuclear pore samples. So as you see here, there's a, I make this a bit bigger, confocal overview of a NUP96 GFP cell line. This was fixed, permeabilized, and stained with a nanobody against GFP. That is working with an imager strand and a docking strand that the imager strand contains paint 660 and a barrier dye optimized for paint imaging. So we take a confocal overview and then we go to a specific region that we're interested in. For instance, here you see the confocal counterpart to the image we are taking right now. And as you can see, there are the green dots. This is a single nuclear pore. And here you see our live view of our MINFLUX measurement right now. So the measurement is running, it's scanning the whole sample, and every time it finds a molecule, it starts this localization procedure Xiaoyu already told you. So it iterated down from a big L to a small L to a final localization. This localization then is added directly to our image. And by this image builds up slowly. And as you can see, for instance, here a nuclear pore, here you see the single proteins that are really visible in this pore. While we image, we see here this blinking window. This is just a detector window that shows us if we have a signal. So at some point you get used to it. If it's dark, it's not localizing. We are just scanning and looking for a single event. And if it's a very fast blink there, then you see that we localize a molecule. When you start this, you get a feeling very easily how this blinking behavior should be. At the same time, you see in our MINFLUX acquisition window always the number of localizations we have, how many traces we measure, how long our measurement was running. I started this before the pause. And you can see how bright our molecules are. All this gives you some information about how well our measurement is running. One very important part, we want to get down to a resolution of 2 nanometers. Or even below that. There are publications out there where we are in the angstrom resolution with our system, where you can see difference between 3.1 and 2.5 nanometers in distances in molecules. But to measure that exactly, you need a very good stabilization. You have to make sure that your sample doesn't move in the time while you're imaging. Every movement there, every small drift will be visible in your data. And for this, we have a special stabilization. We use fiducials, gold beads, that we add to the sample after we stained the sample, after everything is done. We just add the gold beads to the sample, they stick to the ground, and we use a camera to detect the gold beads in an additional beam path with a 980 laser and measure the changes. And if there's a change in position of the gold beads or of the reflection of the gold beads, if there's a change in the reflection pattern, we directly correct this with the piezo's that we have at the system. This is done every 10 milliseconds, roundabout, in this case, every 12. And as you can see here, in orange, we see the stabilization XY, this dot, and in green, you see this in Z. And if I would now start bumping on the table here, I kicked this, I'm sorry, I bumped too hard on the microscope and now I lost the stabilization. Sorry. We can directly change this. I start the stabilization again. You see the gold beads. I take a reference. Now it takes a position, it sees the reflection pattern, and then you see how this normal drift is. Then I can lock it, and then it's stabilized. And here you can see with which stabilization this is localized. So it corrects with a precision of below 1 nanometer. And if I now hit the microscope a bit less hard, but just produce small vibrations here, you see that it also always directly corrects this. So, perfect. Now I stop the measurement. And as you can see here, you see all the nuclear pores very well resolved. They have a diameter here. Oh, sorry, you can directly check with the line profile the diameter of a nuclear pore. And then you can see that it's exactly those 110, 105 nanometers between the upper and the lower ring of the nuclear pore. So now this was just normal 2D imaging. And now I would like to set up a 3D imaging sequence. For this, I just recenter the stabilization. And I start, because I want to take a look at another position, I just start a confocal overview. Sorry. Just let me go to the overview and start this. So you already see here a nuclear pore. I can focus a bit, take a look at it. Till I'm in focus, then I might want to move at a specific position. Yeah. Let's say I'm happy with that position. I take a smaller overview, confocal overview. You can directly copy this in another region and take a look at this. Sorry. Then I focus here a bit. And then I can decide that I'm very interested in, let's say, this field of view here. Get an overview. I put this in green because it's just color-coded now. It's a GFP channel. I changed the settings a bit and the size of the image. So maybe this is a bit too small. I'm interested in this region. I lock the acquisition again, or the stabilization again. A reference and lock. Then I say I'm interested in this region and I want to do a 3D imaging now. Then I just have to go to the MINFLUX acquisition, decide for the sequence I want to use. Before I want to 2D imaging, automatically the system set all settings for the 2D imaging, a 2D donut, which patterns are used, how we iterate down. Now I just decide to use 3D. That's then the imaging 3D sequence. I rename this as MINFLUX 3D. I have to change the acquisition power a bit because with the 3D donut, we need different powers because the power distribution is a bit different. And then I can directly start the MINFLUX measurement. And what you see now is, again, this blinking window. Every time you see a blink, a molecule is localized, and you already see here this live view. And every time we have this blinking, a new molecule or several new localizations are found and are plotted. And here you already see in the beginning there the nuclear pore starts to come. The sample is always scanned from the right top down, and every time it finds a single molecule, it localizes it. And when it's on the left bottom, it starts on the top again. And by this, the image builds up slowly. We will come back in a moment to look at this. Evelyn will take over now and say something about the MINFLUX. However, if you already have a question about how the system works and how you, or to some specifics, you can already ask a question right now.
Okay, so we'll...
I stop sharing.
Thank you. And you should now see my PowerPoint. Okay, great. So thank you, Nichols. I just wanted to show you all the system that Nichols is sat in front of. So this is our MINFLUX microscope. It's a turnkey system, so it can actually be controlled entirely remotely. Everything can be controlled via the computer. We really make an effort to motorize the majority as well of our alignment components. So as long as you have someone to put the sample on the stage, you can run the system entirely from your own laptop. The system is built around a standard microscope body. So we use an Olympus IX83 at the moment. And this includes normal epifluorescence and transmission options, which allows you to search for your regions of interest, just as you would on a normal wide field or confocal microscope. We also, as Nichols showed you, have confocal functionality. At its core, this is just a very good confocal microscope with some extra tricks up its sleeve in order to perform the MINFLUX measurements. We also have in our standard MINFLUX configuration multicolor options. So we can provide MINFLUX lasers in the 488, 560, and 640 ranges. The most important part for our very high precision imaging is that active 3D stabilization system that you were shown. So this is comprised of an additional piezo stage that sits on top of our regular microscope stage. So this allows us to, as we measure, we record those slight deviations from the reference position that we set. We are able to actively correct for those, which means that it's an active process as opposed to something that we need to apply in post-processing, which is what is usually done for single molecule measurements that are camera-based. As Xiaoyu talked to you about earlier, we also have ultra-fast beam scanning and positioning made possible by our electro-optical deflectors, which is what makes it at all possible for us to perform tracking at the speeds that we can. We also have several different upgrade options with this system, so it is also possible to include not only those additional MINFLUX laser colors, but also to incorporate STED lasers within the system. So just to reiterate the basic MINFLUX principle, what we really need to understand about how this microscope works is that we're looking to minimize the number of photons we get out of a fluorophore. We have our single emitter, and if we place our donut relatively close, we get a few photons. If we place it at this position, we're getting more photons. It's experiencing more excitation from this excitation donut. And if we pre-calculate a pattern, so for example, the 3-point pattern triangular or the 6-point pattern hexagon that is part of our system, then that allows us to triangulate our molecule position. And as Xiaoyu's already explained, although more photons does mean more information, it does mean we know more about the position of the molecule. The driving factor that makes MINFLUX different from a camera-based localization technique is that L, that shrinking of our search pattern, or as it's called in the literature, targeted coordinate pattern. So our 6 points are in 2D, again, as Xiaoyu's already explained, but just as a nice visualization, we're placing our donut directly on each of these points, recording information, and then using that information to calculate a position for our molecule and recentering our pattern before we then iterate down to a smaller pattern. So we're continuing to calculate a more and more precise estimation of where our molecule is. Because of course, in a real scenario, it would be lovely if we knew exactly where it was, we could place our donut on top and get no emission. But in a real scenario, we don't know where the molecule is. We need to do some searches first, which is where these large L patterns come in. So we're searching for an emission, and then we continue to iterate down, becoming more and more precise until we produce some final localizations at our highest precision. So we end up with a small bundle of localizations, allowing us to reach those very high precisions. So MINFLUX can be used for both nanoscopy and for tracking. What Nichols is doing right now is nanoscopy. And for nanoscopy, we need to have single molecules. So whatever labeling approach you're using for a MINFLUX imaging experiment, that fluorophore needs to be able to undergo a state transition. So it needs to be able to blink on and off in some way. So that allows us to then localize our one molecule and one by one localize every different molecule within our structure of interest. So the main strategies, the main ways that this is done are photo switching approaches, so using carbocyanine dyes like Alexa 647 or our flux dye range. So these are the most common sort of implementations for direct storm microscopy. You may have come across these before, where you're using a thiol-containing buffer, so something like beta-mercaptoethanol or mercaptoethylamine. And that thiol component can bind to our emitting fluorophore and put it into a reversible dark state. So this reversible dark state is relatively long-lived, which means that the majority, at any given time, the majority of your molecules will be not emitting, and we'll end up with some spontaneous return to the emitting state, allowing us to create that blinking behavior that's necessary for MINFLUX. We're also able to trigger this back reaction with ultraviolet light, the 405. So other examples include spontaneously blinking dyes, as well as photoactivatable or photoconvertible options. However, our two favorites are the photo switching and the one that Nichols is showing you right now, which is DNA paint. So DNA paint works by having transient binding of an imager strand attached to a fluorophore, which transiently binds to a docking strand complementary to that imager strand. So this is quite easy to implement in standard confocal sample preparation. So any kind of primary antibody secondary system can be used very easily with DNA paint. And it also allows you to control the level of blinking very, very precisely by adjusting the concentration of imager that you're including in the system. So what's on the system right now is a U2OS cell line, which is constitutively expressing GFP NUP96. And as Xiaoyu showed you, we're able to resolve each of these subunits in an 8-fold symmetry. This was from the original paper where we described the implementation of MINFLUX that we use for our Abberior system, as opposed to the initial invention. So if you're interested at all in the technical aspects of this system, I would really recommend taking a look at the paper in Nature Communications by Schmidt et al. But we were able to show that with our commercial implementation, we're also capable of resolving below a single nanometer on a nuclear pore sample in 2D. The great thing about the nuclear pore as a benchmark is that it has those two layers. So we're able to use it not only for 2D benchmarking, but also for 3D benchmarking. And we are capable here of resolving these cytoplasmic and nucleoplasmic rings, which sit about 50 nanometers apart from one another in 205 nanometer ring arrays. So we are capable of resolving these with a localization precision of below 2.2 nanometers in all axes. So in fact, we're even a little bit better in the Z-axis, which is significantly different to standard microscopy, given the usual elongation in the Z-axis. It's one of the really nice things about MINFLUX is that relative isotropy. So, of course, today we're showing you a relatively small area so that we can get an image during the time that we're together, but we're also able to image over larger regions. So here we have a full nucleus, imaging the nuclear pores across the whole area. And we see that there's no loss in resolution as you increase the size of your region of interest. So, again, as Xiaoyu's already touched on, there's been a lot of cutting edge research in the last few years with MINFLUX. It's been really wonderful to watch how the technique has developed as the first commercial systems went into labs in around 2020, and now the field is blossoming. There's a lot of very important research being done with these systems. So a couple that I'd like to touch on would be this particular project where we were brought a sample that had been imaged instead, and they were seeing what they thought were large rings in the cell membrane. So this is a protein called NINJ1, which is important in apoptosis. It's a form of cell death. So these customers were able to see a large sort of 70 nanometer ring instead, and they wondered whether there was any substructure that we might be able to resolve in MINFLUX. But they did warn us that they don't always see these rings. It depends very much on the treatment of the cells. So when we first got the cells, we couldn't see any of these large rings. We were a bit disappointed, but what we could see were these much smaller rings. So we were able to resolve these small 40 nanometer rings within the cell membrane that were completely invisible with both STED and confocal microscopy. And what became clear and apparent was that these structures were actually correlating to something that was found with a cryo-electron microscopy project that the scientists were also part of. So we were able to use MINFLUX microscopy, light microscopy, to correlate with cryo-EM findings about the structures formed by these little NINJ1 proteins. Another really nice piece from one of our users is looking at RIR2 tetramers. So these are cardiovascular receptors. So these are known to create tetrameric structures, and it's possible with MINFLUX to resolve these tetramers, which are spaced at a minimum of 16 nanometers apart. So there is definitely a quantitative possibility with MINFLUX if you have good knowledge of the labeling approach that you're using and the structure beneath. Another nice example from the kind of early publications is this ribbon synapse. So while these have been imaged with Storm and Stead before, it wasn't always clear how they were arranged in the Z direction. So although when looked from the side, you can see this nice single layer, when we image with 3D MINFLUX, we are able to see that they're actually two parallel lines sitting very close to one another. So this resolution, this resolving power actually allowed for the creation of a new model of how these particular ribbon synapses function. I'd also like to talk to you about our one of my most favorite papers that's come out recently is the nuclear pore imaging and co-tracking. So here the researchers wanted to both have a very high resolution image of the nuclear pore and also be able to track import and export through the nuclear pore, both of which are very technically challenging, but were both possible with a MINFLUX microscope. So what the researchers did was take living cells, partially permeabilized living cells, and attach a self-blinking dye via a nanobody to our nuclear pore complex, allowing them to image with very high precision the structure of the pore. And then through those imaged pores, they were able to track importing and exporting molecules. So if we look at it from the side, so XZ in this image, you can see things that are passing through those pores, and also things that are not passing through the pores that are starting and then failing to continue. So this is my favorite diagram from that paper. You can see that we have import, export, and also these aborted tracks where things have tried to go through the pore and then failed to continue through. So one of the really interesting findings of this paper was that there was a point of no return at the midplane of the nuclear pore. If a molecule passed that point, it would always continue through the pore. It would never then return to the other side. So this kind of precision would only be possible with a MINFLUX microscope. I'd also like to briefly talk to you about two-color MINFLUX microscopy, and then after this, I'll hand you back to Nichols and we can look at the image that we're taking right now. But here we have, as Xiaoyu spoke about, the nuclear cytoskeleton has these very nicely arranged spectrin rings. Spectrin is not the only part of the nuclear cytoskeleton. There are other proteins such as adenosine and this protein PALM1. So before this work, it wasn't entirely clear how PALM1 was arranged, how PALM1 was distributed within the membrane cytoskeleton. But from this work, it was possible to image in two colors, PALM1 and adenosine, another membrane cytoskeleton protein, and show that PALM1 is actually sandwiched between these adenosine molecules. So if we see a little bit clearer here, if you unwrap that axon, you can see the organization of the two different proteins. So multicolor MINFLUX can be done in numerous different ways. One of those ways is exchange paint. So here we have an example of 3 different mitochondrial proteins that were imaged with an exchange paint approach. We see we've got TOM70, MYC60, and ATP5B, and you can very clearly see the difference in localization of those 3 proteins. The other method of doing two-color MINFLUX is using a ratiometric approach, so spectral unmixing, essentially. So we use two dyes that are spectrally distinct, but both excited with the same laser. So our favorite combination is Flux 640 and Flux 680, as these both behave very nicely under similar buffer conditions. So the way that this works is that our fluorophores are emitting and detected over two different detectors. So, for example, here we have A, which would be more left shifted. That's our 640, and that's being majority received on detector 1. Whereas here, B is more right shifted in its spectra and is being majority detected on detector 2. So we can take those counts that we're receiving on each detector to clearly define where each molecule is coming from in terms of its population, which allows us to do a simple two-color measurement in the same acquisition without having to do post post-alignment, because we're using exactly the same excitation laser, and the spectral shift is so small that the chromatic aberration is negligible. So an example here of, again, a mitochondrial protein. This is TOM20 and mitophilin. And another one here where we see in 3D those very clearly defined two populations. Another favorite of mine is this H. pylori bacterium. So we get this lovely kind of spiral shape. And this was a pulse chase experiment. So it was majority in our Flux 680 with a few different specs of our Sci5, our 640 molecule. So you can see the full structure in 3D. Okay, so before I go on to tracking, I would hand back to Nichols so that we can take a look at the image that he's been gathering at the moment.
Thank you, Evelyn. Again, if there are any questions, please ask or write it into the chat. We are here now with our 3D measurement. It is running now for 20 minutes. And as you see, we already here in the live view already got a lot of different localizations, and we're able to visualize a lot of the parts of the nuclear pore. The live view is just a voxel-sized, in this case, a XY view. You can render this however you like. Every it's color coded for the number of localization that we find in a specific position. We rendered this now with the 8 nanometer size. This is the standard setting for a 3D sample in the beginning, but you could also render with a lower size or a bigger size. At a specific point, one has to ask which is the right size for this. In 3D, we typically get a resolution with the settings and with this size, not resolution, a localization precision of about 4 nanometers. So you could render this with 4 nanometers or with any size you like. The measurement is still running. You still see blinking, some events, and sometimes localizations. And you also see the iteration count that we have in the different localizations. We localized now 600 positions, and every position is imaged several times, so more than 12,000 localizations are gathered already. In Inspector itself, you can visualize this just as an XY overview, or you can take a look from the side in, for instance, in XZ. So now we look and you see this added up over the whole region, or in YZ. And you can see the nuclear pore, the upper part and the lower part. And because the pore itself show the pores itself are all on the same level, but different pores are at different levels, and so there's a slight shift in the. That's why you see this not clearly separated at the different positions. So this is an XY again. You can plot this data so however you like. You can also plot something else than the localizations. You could plot specific characteristics of the image that we have taken. For instance, you can plot the time when this image was taken. Maybe for this we change the color map to a rainbow color map, and then you see the later localizations and the early localizations. You can plot for other parameters that we measure, like the photon count that we have at every specific point. For every localization, you get much more information than I show you right now. Just let me check for a moment, localization again. And all this is stored. And you can later export this anywhere you want and render your data or analyze your data with whatever you like. For this, you can export this as a ZAR file that contains all the information, or also the localization data as NumPy data, MATLAB data, or JSON data. But also, we can directly 3D visualize this live. For this, you can launch ParaView. That's an open source program, and we have a special ParaView bridge that allows us to live render our data. And every time that there is new localization, they are also added directly here. Here you see the 3D data color-coded, and you can start moving it around, and then you can move to a specific pore, and you can see the upper and the lower rings of the single pores. As you see right now, the pores are not complete, so we should image or would image a bit longer to get as much of the pore as possible. Typically, with the settings, we get to a localization precision, a labeling efficiency of 60% of the with the strategy of the possible positions that we can measure. As you have always two, you can get a whole nuclear pore in the end with the old field symmetry in some cases. Okay, I will stop this now and just open a measurement that was running for a bit longer. As you can see here, we also launched ParaView. And this would be now an image of nuclear pores that were measured for about an hour. And as you can see, if you move closer in, it's a clear separation between the nucleoplasmic and cytoplasmic region. And you see that the pores are quite well filled, and the localization precision, they're all separated, and you can separate all the single positions in the nuclear pore. Okay, so just if I would now be happy with this and just start another measurement, I could again just unlock my stabilization, try to find a new cell, do a confocal, define for a region. Let's say I'm interested in this region here. I still can lock this if I don't move it. Take a look at this, decide that this is a fine region, that this shows the structures I want to see. I decide for my imaging sequence, and then I just start the MINFLUX measurement, and another measurement is running, and this will run. Now we find the localizations are plotted, and this will run as long as I want this to run. Okay. Yeah. That's it already. Do you have any questions concerning the 3D imaging and sequence? One important part I should comment on is We use for our patterns this EOD settings for XY, EOD scanner for XY positioning. But for the 3D scanning, we have to use the 3D donut and scan it also in the Z direction. That's why we have a lot of more iterations. So iterations means always this moving step in 3D than in 2D. And for this, we use a DM, a deformable mirror that allows us very fast scanning in Z, just additionally. And the scanners, the EODs are about 100 times faster than the Galvo, at least the positioning of them. For this, we can be much faster in the localization with the big MINFLUX system, as we can call this, compared to the Mirava, which allows us very fast tracking. And I would comment on the tracking after Evelyn gave you a short introduction to that.
Okay, yes, I'll talk to you a little bit about tracking. I'd also just like to emphasize that the complexity of MINFLUX is definitely much more on the side that we take care of. As you can see from Nichols, setting up a measurement takes just a few clicks. So when you have a sample that's running well, the actual operation of the microscope is not particularly complex. And the nice thing is that that's true for 2D and 3D measurements. And much in the same way that you switch from a 2D measurement to a 3D measurement, you can also switch to a tracking measurement just by changing that sequence that you're feeding into the microscope, telling it all of these complex settings, what to do with its EODs, what to do with its DM. All of that is done via just changing that drop down. So tracking is, of course, one of the real cutting edge parts of the MINFLUX. So I have a video here that's a subsection of one of the videos. A video that's not playing, okay. So the video I was going to show you, you already saw, thankfully, in Xiaoyu's presentation, where we're localizing a molecule. And as it continues to move, we continue those iterations as it is moving across your region of interest. So we're continually localizing a molecule. So as long as it doesn't escape our search radius, we can just follow that continuously until it bleaches, or until it moves too fast and disappears out of our catch range. So a really nice example of the top speed that's achievable with a MINFLUX microscope is these lipid bilayer samples. So you can see in this confocal here, it's a supported lipid bilayer, an artificial system, and we've spiked it with some ATO647N tagged cholesterol. So this is zipping around very fast within this structure. There's nothing really restricting the movement other than the contents of the bilayer. And we're able to catch and track individual lipids within this bilayer. So this track takes, so it's played back in roughly real time. It was taken over less than 3 seconds. But within those 3 seconds, we were able to measure over 18,000 data points. So we're using the fluorophores photon budget with the highest efficiency possible in order to gather a lot of information at a high precision. So because of those EODs, we're able to achieve a single data point every 100 microseconds, which is significantly faster than a camera-based tracking technique. With this top speed, we're able to reach around a 13, 14 nanometer localization precision, again, significantly better than is possible at high speed with a camera. If speed is a little bit less important, but still a factor, then we're able to reduce the speed and increase the precision, as we would do for a kinesin experiment where we want to see that stepping behavior. And we're able to achieve up to a kilohertz with 2 nanometer localization precision. So just a couple of cool examples from recent literature. So membrane research is a really strong focus for groups that are tracking. with MINFLUX at the moment. So this group here was able to use that spectral demixing that I spoke about with the two-color approach, to use that spectral demixing in order to track two different molecules and look at their behavior within the same areas of the membrane. So they were able to see the behavior in this case, they were looking at picket fence models. They were looking at corralling and the adjustment of behavior in different sort of membrane subdomains of this tagged cholesterol and a tagged receptor protein. And as Xiaoyu went over in detail, tracking kinesins, tracking dyneins, it's been a real step change in this whole field, where before it was very much limited by camera-based tracking approaches, either having to tag with a heavy gold bead that was affecting the dynamics and the behavior, or by just not having the precision or the temporal resolution that was necessary to optically watch this behavior within a cell. So here, this is a dynein example showing again that kind of stepping behavior that was predicted and seen for the first time with MINFLUX. So if there's any questions at all, please feel free to pop them in the chat. But otherwise, I will again hand back to Nichols, and he will show you a bit of tracking data on the system.
Just a moment. Okay, thank you, Evelyn. Yes, MINFLUX tracking. I just want to show you a short example how the data looks and how we would typically track. For tracking, it would be the same as for imaging. So in this case now, I had a sample that was a lipid bilayer. And in this lipid bilayer, this was stained with star green, a lot of lipids, and a few lipids were stained with ATO647N. And this is what you can see here, an overview. It's just a layer, and in this layer, there are single red dots that diffuse freely in here, somehow freely. It looks like this. And if I would now have taken that image, I would just decide for a region I'm interested in. Again, define this as my MINFLUX region, and then I would just take the sequence I need. Instead of an imaging sequence, I would choose a tracking sequence, and there are different tracking sequences, tracking for 2D very fast, tracking 2D for slower events, and also tracking for 3D events. So for diffusion in 3 dimensions. I start the measurement, and then we will always, this system will scan as it did for the imaging, and always when it finds a molecule, it will just follow it. It will reposition very fast with our EODs and the Galvo. And as Evelyn already said, it can be done with a speed of about 100 microseconds per time point, meaning we are 100 times faster than any camera-based system. And we get down to localization precision when we are very fast of a bit above 10 nanometers. If we say we take more time for localization, we, let's say, not 100 microseconds, but 1 millisecond, we can go down to localization precision of a few nanometers, about 2 to 3. This depends very much on the speed of the structure we want to take a look at. And it depends on the dye that we are using, the brightness. In the end, concerning the speed, we are limited by 2 things. One is how fast we really can follow that molecule in the small steps and how fast we can localize. And this has something to do with how fast we can scan. But the other reason is we are also limited by the photon output of our dye, because we have to gather in 100 microseconds already tens of photons, 20 at least, and this means we already need a very high photon output of the dye. So for tracking, I always suggest taking bright dyes. what you can see here, so what you also saw, you do not need necessarily, especially for tracking, not necessarily switching dyes or dyes that go into an on and off state. It's also, maybe it's even better to have constitutively active dyes that are always that are in a low concentration. So you can always, in your donut, always have only one dye and can track this dye. We have, when we start this localization, we always have quality control checks. Do we really have a single molecule? We can define this by analyzing the pattern that we have. And if we have a single molecule, we track this single molecule then, as long as we can track it till it's bleached or going to a dark state. Yeah, as I said, the speed is about 100 microseconds. And here you see a typical result of this. In Inspector, you see color-coded always a single trace ID. So every point is a localization. And if it's the same color, it belongs together. To visualize this as a trace, to really follow single molecule, you can click here on Compile Trace Data and visualize the data then in ParaView. I would go to this. Here you see that an example in ParaView. It's also color-coded in the same way. And if you zoom a bit in, you see the different molecules and how they behave. You can get a lot of information. You can get the information of all the single localizations. If you go here, for instance, the point data, you see a list of thousands of points with the exact timing, the exact localization, the exact information, photon counts that you had at this point. But you also can analyze this for the single steps from one point. Just a second, I will show also the localizations for a moment. Yeah, sorry, I will come to that in a moment. Cell data, or you get the information of the whole trace. Every trace gets a number, and you can, for instance, take a look at the traces that have the longest distance from start to the beginning, that have the longest trace duration, or the ones with the size. For instance, trace 1935 is the trace with the longest, with the most single positions that we measured. And then you could go, so you have to see all the traces here. But we could also go to extract the blocks, decide for this trace, and take just a look at the single trace. And there you already see how differently this trace behaves. So this lipid is diffusing very freely, but at some point it gets somehow stuck at positions there, probably lipid rafts or something else. And this is things that probably are not visible if your time resolution is not the right thing. If you would measure just every 100 or 1,000 time points with a camera-based system, you would not be able to see that they are stuck at specific positions. You can visualize also all these different points. For instance, you can take a look at the speed. And what you see in blue, this is always points where the molecule is kind of stuck and moving not very fast. And when it's moving faster, the traces are colored in yellow or in red. And you can extract all this information and analyze all your traces. And interestingly, you can also analyze this then for the, if it moves differently in different parts of cells, if the molecules behave differently when they are closer to something different. So you get a lot of the highest resolution, spatial and temporal, about the behavior of the protein or a lipid or a dye in a living cell. Okay. That's it from my side. So if you have any questions, we would be happy to answer any.
So I think there is no questions for now, but you can always write to us and we can arrange to get your questions answered from the experts like Evelyn or Nick or Bastian.
Well, thank you very much all for your time. Thank you for letting us present to you today.
Thank you. And if you have any questions, you can always contact us. We are happy to help.
Yeah. Thank you, Nick. Thank you, Evelyn and Xiaoyu. This was a wonderful wonderful session, and we got to learn a lot. And we'll also feature this session on our website and our YouTube channel so that people can see and learn and get more details about this super resolution techniques, which is now different from regular super resolution technique, which is very much popular till date. But since we are with optical systems, we are able to get the resolution which is at par with the electron microscope systems. So maybe people will be more interested to learn about this technique, and I'm hoping that people will reach out to us.
Okay, thank you very much. Thank you to everybody. Yeah.
Thanks.
Thank you.
Okay, see you. We close
Bye.
this. Bye-bye.
Bye.
Thank you. Bye.